we are talking about integrated nutrient management. Okay, so integrated nutrient management means integration of all the possible nutrient sources to your nutrient management schedule. Now that is basically uh, means what you can say. If we talk about integrated nutrient management, that means we are talking about the sources of nutrients. But what about the time? Means means when to apply and where to apply. So this particular approach of nutrient management actually takes care of all these things, all these aspects. That is site-specific nutrient management. So, as the name is already self-explanatory, that here it's a dynamic field-specific nutrient management protocol where we actually see how much nutrient is present there as an indigenous nutrient supply, how much nutrient is coming through crop residue and manure, maybe how much is coming from irrigation water, and then we fix a target, a yield target, how much yield we need to have, and for attainment of that much yield, how much nutrient we have to give as a fertilizer. And this comes under precision nutrient management, which is also which is also a part of precision agriculture. That is the need of today's agricultural scenes in India. So some principles of this site-specific nutrient management we are going to discuss today. Now, in this regard only, the International Plant Nutrition Institute, this IPNI, they have started one way back. They started one nutrient stewardship, 4R nutrient stewardship. That is right product, right rate, right time, right methodology. Means you have to use the right product. Means for certain soil, certain products are important. Means that basically more tells about the integrated nutrient management. All the nutrient sources you need to exploit. Then it has to be applied in right rate. That means we have to see how much nutrient is required based on the soil test or based on the site plant testing, based on site specific nutrient management theories, how much is applied. Then right time, that is when to apply, means when the nutrient demand of that crop will be maximum. There will be maximum chance of utilization of the nutrient if we apply at that time. Maybe we can measure the nutrient demands, mostly nitrogen demands. Real-time nitrogen monitoring we can do by leaf color chart or maybe green seeker. So accordingly, we have to apply our fertilizers at those times only. Then right methodology, how to apply, means we have talked about this also. Say we can apply by spraying, or we can apply by basal doses, maybe soil application. If we are applying like phosphorus fertilizers, we can go for band placement, or if we can go directly to the root zones through subsurface irrigation. So that is a good way of applying nutrients for better nutrient use efficiency. So that is the 4R principle. So all this actually comes under this site specific nutrient management. So then according to that, let us talk about, means we are basically talking about how much nutrient we have to apply and when to apply, how to apply. So there are certain approaches that are being followed right now in our country for making fertilizer recommendation to the crops. First one is general recommendations. So every state has recommendation for fertilizers for every crop in their area in a particular season. Every state, means state of Maharashtra also have the NCR region here in Delhi. They have one particular state recommendation for every crop like rice, wheat, maize, everything. So that this recommendation actually are developed by on the basis of multi-locational field experiments conducted with graded doses of fertilizer nutrients individually and in combination for a long term. Actually, so from those trials, some fertilizer recommendations generally come up, and those are generally serve as state recommendation, a blanket dose. But obviously, site specificity or precision is not there. So then, to approach that precision, uh, there is some other types of nutrient management practices also. Uh, we can do that on the basis of fertility ratings. So in this approach, what we do, we basically test our soils, maybe for organic carbon, phosphorus, potassium, nitrogen, and those values are then divided into given different fertility ratings. Say for organic carbon, the medium rating is 0.5 to 0.75. Similarly, for phosphorus, some values are there. And if the soil sample which is being tested, that particular sample is within that medium range, then we can actually apply the state recommended dose. But if that soil fertility rating falls in the low range, then we need to apply 25% more of that state recommended dose of that particular nutrient, obviously. And if it is coming in high fertility rating, then we have to apply 25% less of that state recommended fertilizer dose. So this is followed. Now, based on this only, this nutrient ratings, fertility ratings, uh, another thing has been developed is nutrient index. This is developed by Parker way back in 1951. This is basically useful for calculating the fertilizer recommendation approach for a given area, like a village or a block or maybe a district even. So here we take the samples. We again analyze those samples for different parameters, and then we can know how much of those samples falls in low, medium or high category. Then this nutrient index can be calculated as this formula, means low into 1, medium samples into 2, and high samples into 3 divided by total number of samples. So where just NLMM and NH are the number of soil samples falling in low, medium, high category, and NT is the total number of soil samples analyzed. Okay. Then if this value of nutrient index is less than 1.5, then that fertility of that nutrient in that particular area, it can be categorized as low. If it falls between 1.5 to 2.5, then it's categorized as medium, and if it is more than 2.5, then it can be categorized as high. And accordingly, we can recommend fertilizers. Means if it's a medium, then we can just apply the state recommended dose. If it is less than that, less than 1.5, then we can use 25% extra of the fertilizer nutrient doses, means as compared to the state recommended dose. Now you see, if all the samples generally falls in low category, then the value will be 1, and if all the samples falls in high category, then this value of Ni will be 3. So here a large weightage has been given to the medium category, means 1.5 to 2.5, whereas for high samples only 2.5 to 3. So that's why Ramamurthy and Bajaj in 1969, they actually modified these limits as 1.67 to 2.33 for medium range soils. So that can be followed, and it is useful for developing fertilizer recommendations for a blocks or maybe district level or maybe a village level. So here also some generalization is there, but obviously it is better than the state recommendation. So some precision or site specificity is there. on this nutrient index approach. Then another method or concept of fertilizer recommendation is based on the critical limit of soil available nutrient. This concept has been developed by Keaton Nelson in 1965. So here what we do, now this critical limit is the value of soil available nutrient above which the probability of getting economic response to application of fertilizer nutrient is very low, while soils having the value of soil available nutrient below the critical limit, the probability of getting economic response is very high. So that means if the soil is having the nutrient content more than that particular critical limit for that particular nutrient, then the soil is not responsive enough economically. Means if we again apply some nutrient, say for zinc, if the zinc is having value more than the critical limit, the soil test value, then even if we apply zinc, it will not affect the yields in a great way. So that is basically critical limit. So how it is generally obtained? We do some field study with some soils which is having an inherent fertility gradient, or we can create that fertility gradient obviously by using the particular nutrient in a graded dose in that field in some plots. So then we can develop this experimental field which is having a fertility gradient, and in that fertility gradient then we apply our treatments which is having also, like say if you are doing for phosphorus, then it will be zero phosphorus, then some graded doses of phosphorus. Then we take the brace percent yield of all those plots, and also we take the soil test value, means we test the soil for that particular nutrient, say for this example phosphorus. So now that's brace percent yield or the relative yield. It is basically the ratio of a particular treatment yield obtained without that particular treatment, means yield obtained in the control, and divided by maximum yield obtained with that particular nutrient, means in that treatment only how much is yield, how much yield is obtained, and that and yield obtained in the control, that is yield obtained without particular nutrient, so multiplied by 100. So that is what we know as the relative yield or brace percent yield. Now that yield is also having a corresponding soil test value of that particular plot. So those are then plotted in a graph paper, and as we can see, we can clearly create 4 quadrant here, keeping in view that maximum number of points should fall in the first and the third quadrant, since it's a quadratic kind of relationship. So maximum means we can move these lines, right? And generally this horizontal line actually falls around 85% yield percentage level, and then we can move this line to a point that maximum amount of this points falls in the first quadrant or in the third quadrant. Then we see where this is falling on this x-axis, in which point it is intercepting. So that point is basically the critical soil test value. Now if for this is for phosphorus, so if phosphorus soil phosphorus is more than this value, then there should not be any application of phosphorus, because it is less than that critical limit. And if it is less than this critical limit, so there if we apply phosphorus, then there will be increase economic viable increase in productivity and yield. Now this is generally, this limit is critical limit is not generally applied for the major nutrients, which is actually required in large amounts, but for the micronutrients where we have to make the decision where to apply or whether to apply or not to apply, because it's required in very small amount, there this critical limit approach is much successful, and these are the critical limits and fertilizer doses for different micronutrients like zinc, iron, manganese, copper, boron, and molybdenum. Uh, then another nutrient management approach is according to the Michaelis-Bray concept. Now, as we know, Michaelis is the first one who actually quantified the increments in yield by uh, application of a single growth factor or single nutrient. Now he stated that yield can be increased by a single growth factor even when it is not present in minimum, as long as it is not present in optimum. So that's Michaelis law of physiological relationship. And an increase in the yield of the crop as a result of increasing a single growth factor is proportional to decrement from the maximum yield obtained by increasing that particular growth factor. Now that's Michaelis growth law. So here actually what he is doing, he is quantifying that increase in the yield by increasing one single unit of the nutrient, means one single unit supply of one unit of nutrient, or say fertilizer. That is growth factor, what it termed as. So if that increase is dy by dx, where y is the percent yield and x is the dose of fertilizer nutrient, then it will be equals to a minus y into c. Now this a here is the theoretical maximum yield, minus y is the current percent yield, into c. Now c is the proportionality factor here, efficiency coefficient of fertilizer form of that nutrient, means how much the plant can take up nutrient from the fertilizer that is being applied. So if we integrate this and then take the log, it comes around log a minus y is equals to log a minus c x. So that is the relation. But in this actually what is not covered, that is the source of indigenous nutrient supply, means what is they are already present in the soil and plant can take up from that also. So that concept Bray introduced in 1945, that he stated that plants utilize nutrients from both soil as well also from the fertilizer source, and growth of the plant is dependent on utilization of both the forms of nutrient, right? So therefore he just modifies this Michaelis equation as this one, which is the Michaelis-Bray equation, where he added one factor, means log a minus cx minus c1b. This b is the soil test value and c1 is the efficiency coefficient of soil form of nutrient. Okay, means how much actually plant can take up from the nutrient, that specific nutrient available in means actually inherently available in that soil. So that is C1B. Cx, x is the amount of fertilizer applied, and C how much Cx means the total amount of fertilizer nutrient the plant is able to take up. So this is the equation, and if we on a particular soil, when we are growing a particular crop and for a particular nutrient, so we can actually calculate this C and C1 from those experimental plots. And theoretical maximum yield also we know, and also can be calculated by selecting the low, medium and high yields from the curve of yield versus applied fertilizer nutrient. using this equation actually. So then this we know, and this log A also we can know. Then we can actually calculate how much fertilizer, means how much of this X is required for reaching of for getting a particular yield. That is why. So that we can do through this concept. Uh, then as proposed by Ramamurthy in 1974, we can also uh, get the fertilizer recommendation for a particular crop in a particular type of soil. So through this multiple regression approach actually. So here what he did, so they are also first we need to develop, means all this studies actually comes under soil test crop relation approaches. There we have to first we have to take a field, develop a particular fertility gradient in that field, and then we have to apply a graded nutrient dose as treatment, right? So from those long-term trials, maybe we can develop significant relationship between soil test, fertilizer doses, and crop yields. And uh, and this is somewhat that equation actually looks like. Means if you see the full equation, where this y is the crop yield, then Sn, this A is the intercept obviously of that quadratic kind of graph. Sn, Sp, Sk, then Sn square, Sp square, Sk square are linear and quadratic terms of soil NPK. Now this can be actually developed for different nutrients. Means if it's for nitrogen only, then Ya and all the N terms will be there. Then there is fertilizer terms, Fn, Fp, Fk, Fn square, Fp square, and Fk square. These are the linear and quadratic terms for fertilizer NPK. And then there will be some interaction terms of soil and fertilizer NPK. And these are the coefficient of different terms, this B1, B2, B3, etc. Now, this multiple regression approach can only be applied or can only be viable if the signs of that linear, quadratic, and interaction terms of fertilizer nutrient observed in this multiple regression equation approach is plus, minus, and minus. Means basically, if this kind of signs will be present in that equation, then only it will be a quadratic kind of relation, right? And then only this multiple regression approach can be applied for recommendation of fertilizer doses. Now, first from this kind of trial, this kind of this kind of equation can be developed, and then for calculation of fertilizer dose, which is to be applied for maximum yield, we can use this formula for that particular region whose soil has been used for carrying out this kind of study, this kind of STCS study. So FD, that is FD for maximum yield, that is coefficient of the linear term divided by 2 into coefficient of the quadratic term minus coefficient of the interaction term divided by 2 into coefficient of the quadratic term into soil test value. Now that means these are the say for nitrogen. So this is for the linear term and this is for the quadratic terms that we can have. And if say we need to have a fertilizer dose for economic yield, means for optimum yield. Then with this equation, we add another factor, that is 1 by 2 into coefficient of the quadratic term into R. So this R here is basically the ratio of cost of 1 kg of nutrient to the cost of 1 kg of grain or economic produce. Just 1 second, excuse me. Okay, sorry for that interruption actually. Let us continue. So if we know that R, which is basically same for a particular region, and we have the equation, and then by only selecting that soil and testing that soil for particular nutrient, then we can know that soil test value. Then we can recommend the fertilizer dose, and this is there is site specificity in this approach, because we are treating a particular soil, a particular crop for a particular yield level, and also for a particular region for a certain amount of economics. So that we can do. And then the more advanced one, or we can say more precisive nutrient management can be done through targeted yield approach. This is initially advocated by Traub in 1960, and then also popularized by Ramamurthy and his co-workers in 1967. This is started for fertilizer recommendation of different high yielding varieties introduced during the period of Green Revolution. So for targeted yield, actually, same kind of experimental setup we require, and from that experimental setup, what we need to attain is nutrient requirement in kg per quintal of grain production. Means that we can know. Means for a certain crop, certain variety, how much nutrient it needs for a particular unit of grain production. Then percent contribution from soil available nutrients, means how much nutrients that soil is having, and out of that total nutrient, how much can be taken up by the plant in its growth period. Then percent contribution from fertilizer nutrient, that is CF. That means how much fertilizer we are giving to this means that particular crop, and from that particular amount of fertilizer, how much percentage it can take up. So that is the different parameters we need, and there is one AICRP on STCR, which is the headquarters are situated at Indian Institute of Soil Science, Bhopal. We also have one center in our division in New Delhi. So they basically try to develop these three parameters over the years for a particular crop, for a particular region. Okay. Now, how that can be developed? Uh, Now, earlier, all these parameters are generally developed for normal fertilizer, means where only inorganic fertilizers are used. But nowadays, we also have the equations for which is developed along with the contributions with FIM. So for developing any nutrient, means for developing nutrients, how much is required from every nutrient source. This kind of study is needed. Now, nutrient requirement can be easily measured by digesting one plant sample after its maturity, means after its harvest, and we can actually see how much is the total uptake of nutrient divided by just the grain yield of that particular plot. So we can have nutrient requirement of that particular crop. Then we need to calculate the percent CS, means contribution from soil nutrient, soil pools of nutrient. So that is to be calculated from the control plot, where say this is we are developing for nitrogen. So there will be a plot which is a nitrogen omitted plot, zero nitrogen plot, control plot. So there we have to first see how much is the total uptake of nutrient from that control plot, that we can easily analyze by digesting the plant sample and seeing the nitrogen concentration in the plant parts, divided by the soil test value of that control plot only. So that we can see, and that is the percent CS, means percent contribution from the soil nutrients. Okay. Then we need to calculate.
This meeting is being recorded.
Then we need to calculate. Is it okay? Then we need to calculate the percent CF, means the percent contribution from the fertilizer source. So that actually we need to calculate from the treated plots, means where the particular nutrient dose has been applied. So there we know in that plot, means in that fertilizer applied plot, how much uptake is there, that we know. Then we have to deduct the soil test value of the treated plot for that particular nutrient, say for nitrogen. So means that soil test value into the percent CS by 100. So this portion here actually tells us that how much nutrient is supplied from the soil part, and this here is the total uptake of nutrient, means the total nutrient taken up by the crop, okay, minus how much portion is supplied by the soil part of the nutrient, okay. So that means what is left, this minus this, is equals to how much nutrient is plant is deriving from the fertilizer applied. Now that divided by fertilizer nutrient applied in treated plot into 100 is the percent contribution from fertilizer. So this is how we can actually develop NR, percent CS and percent CF. Okay. Now this is obviously for without FIM, and this parameters can also be developed with FIM, that is percent. There one extra will be added, that is percent contribution from FIM. So this can be this value can be obtained from a plot which is a control plot, but that is FIM is applied, but no external fertilizer is applied. So there we can have the total uptake of the nutrient from control plot with FIM minus the STV of that treated plot with FIM into percent CS by 100 divided by total uptake of nutrient. So there we can see how much percentage of nutrient is being derived from the FIM there by the crop. And from that we can also calculate the percent CF. And from that we can actually have this fertilizer dose calculation, which is NR into 100 divided by percent CF into T, means T is the targeted yield. Means this can be developed, means first if the equations are developed, then this can be, means here this STV will be different for different soils in that region. If we know the crop, its nutrient requirement, already we have known from different experiments, say for wheat. So then NR we know, and the T is the targeted yield there, means how much yield target the farmer is hoping for. So NR into T into 100, that means the total nutrient requirement of that crop, right? Minus CS into STV. If we do now, minus percent CS into STV means how much that soil is capable of supplying the nutrient to the plant. So this total nutrient requirement minus the CS into STV, that is the total amount of nutrient that is to be delivered through fertilizers. Now that fertilizer, that nutrient requirement divided by percent CF, this is the efficiency of fertilizer material for nutrient supply. Then we can have the total amount of fertilizer that needs to be applied for that particular crop, for that particular area, for that particular soil test value, for that particular season even. So this is a kind of precision nutrient management, and site specificity is there. And this kind of adjustment equations also is developed for where FIM means farmyard manures are also being applied. So here you can see this one extra term is there. Percent means here we actually deduct the amount of nutrient that is that can be supplied from the farmyard manures. So we need to apply fertilizers deducting that value. So in this way, what we are doing, we are feeding the crop according to its need. Means how much fertilizer it needs. So that much we apply. So then the nutrient use efficiency also will be higher. No losses will be there. So these things can happen if we use this kind of site specificity approach. And this actually ensures the maintenance of soil fertility, and we go towards a more sustainable kind of farming. Soil health will be maintained, and most importantly, pollution aspect, that the pollution with soil pollution, water pollution, eutrophication, those kind of stuffs we can actually avoid. And by doing this, it can also help the farmer by curtailing some of the economic load on his shoulder. We can cut down the use of fertilizer, which is generally not taken up by the crop, which is generally lost if we use a higher side of this nutrient dose. Now, those are basically the principles of site-specific nutrient management. All those are the principles. Means we need to test the soil. Two things is important. We need to test the soil, means how much is the requirement, and how much is the plant yield target, means how much plant needs to take up and how much is supplied from the soil. So based on that, the rest amount is to be given through your fertilizer. So that you only need to apply. Now, based on that principle, IPNI developed a very good decision support system, that is Nutrient Expert. It's a software, open source software. Obviously, it is freely available in the IPNI website. This is a decision support tool for precision nutrient prescriptions. So all of you actually can go and download that, and it's very easy to use. So here, this software actually gives, it's an Excel based software even. So this software actually gives recommendation for a location based on attainable yield and yield responses to NPK determined from experimental data and estimated using site or those farming information. And the algorithm that has been used in this software, that is obviously derived from the STCR trial data. And the relationship between the balanced uptake of nutrients and grain yield is predicted using this Squares model here. So they have this nutrient expert for rice, hybrid maize, wheat, and soybean, and some other crops also. So this actually, say for feed, there are certain information this decision support system actually needs. So first it needs the what is the yield target of that particular area, means what is the area size, what is the name of the location, means which location it is having, and how much is the yield target is needed, and then generally how much fertilizer is needed, means in the previous years, or also there is the current farmers practice, actually farmers fertilization practice. Then it asks about the means targeted yield there, if there is some organic manual application, if that organic manual application will means give it some residual effect of that, and uh, then it asks about the soil test values, means if those soils are tested, and what is the soil test values of that particular field. Then by giving that means if we can provide this kind of information, means if farmers provide this kind of information, then it will also ask about the sources of fertilizer which is being available there. So based on that, then again it can give a particular fertilizer recommendation dose for a particular crop, for a particular area, for a particular yield target, so that it can give. But then it asks about the profit analysis, means that the last part, means what is the means seed rate and cost of seed, means what is the input cost there, and also what is the output price, means what is the price of wheat, how much that wheat is sold in the market per kg, and the support minimum support price maybe. So by taking that information, then hello, if you can just mute yourself.
Hello.
Yes.
I think.
Should we continue?
Okay. Continue, sir. Continue.
Okay. ना slides move कर रहे हैं ना सर?
Yes, definitely.
Okay. So anyway, that is about the nutrient expert. It is available, freely available software on the IPNI website. You should check it out. Very easy to use, and it is helpful for site-specific nutrient management, giving recommendation to the farmers. Then that was about that nutrient expert. It is about the site-specific nutrient management, means how much to apply to a crop according to the yield target, soil test values. Now, if we say now, if we come for means when to apply and how much in a real-time basis, means nutrient expert also it will give you recommendations pre-sowing, means based on the means the nutrient recommendation you will have before sowing of the crop. But for nitrogen, means nitrogen is one nutrient where real-time nutrient management is required, because if we use your all nitrogen requirement at the this stage, means at sowing, then maximum of that will be lost. So nitrogen requirement we need to give according to the plant's real-time nitrogen demand. So we know that nitrogen requirement of plant, it can be seen visually through the leaf color, leaf chlorophyll content. Means if it's paler, then the chlorophyll development is less and it requires nitrogen. If it is green enough, so then nitrogen is sufficient. We don't need to apply nitrogen. So based on that, one index is there. Based on this approach only, one index is there that we all know, that is normalized difference vegetation index, or NDVI. Now here what we do, here one sensor is there. The trade name of that sensor is Green Seeker. It is this photo is for one handheld one. Then one bigger one is also there. So what the sensors actually do, this actually throws measures uh, the reflectance in the NIR bands and also reflectance from the IR red bands. Okay. Now, what happens if there is a good amount of this infrared, means a good amount of chlorophyll is present, then uh, the red light gets absorbed. High amount of this red light gets absorbed, and thus the reflectance will be very less. Now, what it reflects, obviously, it reflects the green light that we can see, but it also reflects a much higher amount. It actually reflects as compared to the green in the near infrared range that we cannot see actually, but the vegetation strongly reflects that infrared region light, right? So if it is having a more green color leaf, then the NIR values will be higher, right? And if it is a less kind of green color leaf, means yellowish color is there, yellowing is there, then that NIR value will be less, and the red value will be more, because red will be reflected more in that red range. So that is what basically this crop sensor measures, and based on that it gives us an index that is known as Ndvi, and it always ranges from -1 to +1, this Ndvi. Now, based on that, means we can take the green seeker readings of a particular crop, and those readings taken by that green seeker can be used to predict real time in recommendation. Now, it actually results in a more efficient use of nitrogen, and it is a benefit both on the farmers economic point of view, soil point of view, and also from a environmental pollution point of view. So nitrogen losses will be less if we apply according to the plant need. And now for means for development of this kind of fertilizer need in requirement based on this GS values, we need to first estimate the in-season estimated yield that we can have from that NDVI of the test plot divided by the number of days from sowing or transplanting to sensing that sensing of that green seeker values. So that INSEE we can calculate, and GDD also means growing degree days. Also we can calculate minus T from this formula, where T means minimum temperature plus means it's average temperature minus the base temperature. So that we can have. Okay. Then we need to calculate the yield potential with no additional fertilization that we can calculate. Based on that, we can actually also calculate the response index to fertilizer nitrogen. So whenever we are developing a recommended fertilizer recommendation from this means for with this NDVI values, we need to maintain one enriched plot also, means where more than maximum recommended dose of fertilizer nitrogen has been given. Say for NCR region it's 150, so we need to have one plot at 180 kg per hectare dose. So from that, this response index can be calculated, and from that YP0 and response index of NDVI, we can have the yield of the test plot achievable by applying the additional fertilizer nitrogen in. That much yield we can apply, and from these actually fertilizer end dose can be developed for particular crops. Now Vijay Singh and his co-workers actually developed for irrigated wheat in one of their paper in 2011. They developed a particular factor, this 1.85, that comes from this studying this pattern for a longer period of time. So they developed this equation. So this equation is applicable for an entire region where they have conducted this study, for that entire region. So now we have, now based on that equation, now we can, if we take the green seeker value and put that value in all these parameters, then we can actually calculate the fertilizer end dose. Similarly, if we also have the fertilizer end dose for transplanted irrigated rice, also by Vijay Singh and his co-workers, developed in 2000, published in 2015. So based on that equation, if we can take the green seeker value of a particular rice crop in a particular field in Punjab maybe, so by applying that value to this equation, we can actually have how much nitrogen to be applied in that particular stage. So this is more precision. More precision is there. That is sensor-based approach, and more precision we can means we can have through this nutrient management approaches. Now, obviously, that is a more what you can say, that green seeker is not, it is not affordable by all the farmers. And it's a bit of a complex, obviously. Similarly, span meters are also there. So that is also a bit complex for using for daily use of a farmer. So for that, the EV actually developed one very good leaf color chart, which is generally different shades of green, right, on a chart. That is mainly developed by for rice by International Rice Research Institute in Philippines. And similar kind of this leaf color charts had also been developed by the research NRI, National Rice Research Institute in Cuttack for rice, and also for rice, maize, and wheat by Punjab Agriculture University for the adjacent areas of that Punjab, Haryana, and this NCR also. So now this is a also very easy and cheap, easy to use and cheap decision support tool. So for means these recommendations actually have been developed. See, ED actually developed this recommendation. If this LCC readings has to be taken at weekly intervals, starting from 15 day after transplanting, until the initiation of flowering stage. And if LCC values is less than 4, then crop dressing of 30 kg of nitrogen had to be applied in that particular day. So that's very easy. But for development of that recommendation, they have actually undertaken a trial with some graded nitrogen doses, and they have came up with this particular recommendation. So for a rice, if LCC value is less than 4, now what is this 4 actually means? See, here is 6 strips. So each of the strips is assigned a value: 3, 3.5, 4, 4.5, 5, 5, like that. So if it is, say, see, this may be 4. So if the value of leaf is less, falling in this, means less than this, so top dressing of 30 kg of nitrogen has to be done in the same day. For wheat also, means if LCC take the readings at maximum tillering stage, if it is less than 4, then top dressing of 46 kg of nitrogen has to be applied. If that LCC value is less than or equal to more than 4, then top dressing of 20 kg of nitrogen per hectare. Now, obviously, this is just an example for a particular area, for a particular crop. It has been developed for a particular variety also. Now, these readings are there. This LCC based nitrogen recommendation protocols are there, available for rice and wheat, mainly for maize, rice, wheat, and maize, and available in public domain in different literatures. It is available. So it can be used and can be popularized by different particular institutes in their respective regions, I guess. And same research has been carried out in IRI also, in our research group also. So we some mobile based real-time nitrogen management word. So what we are doing here, we are basically converting this LCC to a Android based application. So what we can do, we can take a picture of with our Android smartphone that is available with every farmer, I guess, in nowadays, in today's modern era. So one can take a photo with their mobile, and the inbuilt software will do the all the image processing and give us the RGB values, and which will then correlate with the green seeker based recommendation or the leaf color chart based recommendation of that particular field for that particular RGB value. And it can give the fertilizer recommendation, that how much fertilizer it needs to apply for that particular stage if that much greenness is there. And then it can ask for means, since it's Android based application, we can all means it can ask what is the fertilizer source in that particular area, and based on that how much fertilizer, how much area is having, so that it can give to the farmer. So that is more of a specificity, site specificity, more of a precision in the part of real-time nitrogen application. So that is all about, I guess, site-specific nutrient management. So if any questions regarding to this subject, we can have it.
Participants are requested, if any questions or queries regarding the topic, you can ask directly to sir. Is there any question regarding the topic? Please explain GDD, sir.
Basically, GDD means the growing degree days. So that you can also means it is basically calculated by this formula only, based on what is the temperature of that particular season. So based on that means how much means, as the name suggests, the number of days that is basically provided for that crop growth, means crop needs. So that is how actually we develop this green seeker based equations, adjustment equations.
Next.
Anything else?
Yes, anybody? They can unmute themselves also. Okay, I think they're.
So, sir, I would like to take another just 5 to 10 minutes, since it's a batch which is the students are also from the say UG last year or
Yes,
MSc,
yes.
means PG.
You can.
So
yes.
I would like to take some 5-10 minutes. Means I guess they are preparing for different exams.
Yes.
So,
Please continue.
yeah. So all this information what I have taught you, it's from different books. They are available, and I wanted to just sensitize you, the students, how to read and some new things that is basically not available in the book. So that's what I tried to cover. But you can have all these things by these different books. So let us just talk about the preparation techniques. I think that you guys may be interested in. So if we talk means if we how to go about preparing for different this national level exams, be it JRF, SRF, or maybe ARS. So first we need to identify the basic books according to syllabus. Means if we say if you see the syllabus, I don't know for agronomy, I'm a soil scientist, I know the syllabus for soil science. So if we see the soil science syllabus, then we know the basic book, the most important book is the book that is developed by Indian Society of Soil Science, the Fundamentals of Soil Science, or the Introduction to Soil Science, the new book one. So there all the syllabus is covered. Now as for all the agronomy people here, you guys also might must have some of that book, which is the basic book of that your particular field of study. So you identify that basic book first. You have excellent faculty over there, you consult with them, you consult with your seniors, and first try to identify that, and start with the easy chapters first. Do not go for the very complex ones. Say for soil science, this nutrient fertility, nutrient management, these are more or less easy chapters. So we need to start with that. We should not go first in the clay mineralogy kind of thing, or some soil chemistry. No, first start with the easy chapters. Here the perseverance is the key for this preparation. First you study, start your preparation, some less hours per day, maybe 2 hours, 3 hours you can study, but try to increase that daily studying time over every day. Means first I am studying like 3 hours for 2 weeks, then in the next week I need to make sure that that studying hour for that particular exam needs to be 4 hours, then 5 hours, then it increases, and then it will be a long term. Means at least 1 year you need to prepare for this exam if you need to give a seriously attempt this exam. Then revision is the key obviously here, because you study, then obviously you will forget, then again you revise, again repeat. So this thing is important, and for some exam general agriculture also is important, which generally we need to forget. And as I mentioned also, that revision is the key here, the perseverance with each time you study. Now that revision is also need to be very tricky, I can say. Now as I mentioned, first you need to see that basic book, which is your basic book, study that. After completion of that, you see what are the other books you need according to the syllabus. See, the syllabus is god here. Syllabus is the boss. You need to cover all the points that is written in that syllabus, all the words, all the topics. Now one book actually covers around 50-60% of that topic that you need to see. Then you need to look for the other topics and which books can that be covered from. So now see, if I am studying the preparation, first I am studying the like fundamentals of soil science, reading this, and after once means in the first attempt, that fundamentals of soil science, it need at least 3 months for complete reading of that all the material. Then I need to move to the next book, the associated some topics from some other books. So then I need to do two things at once. We need to see the other books for the additional chapters, and also I need to start revising the fundamentals of soil science. Okay. Now that revision time you need to decrease. First, the whole book, it takes around 3 months maybe for a UG student. But in the first revision, one chapter may be completed like 1 day, 2 days, and that revision time needs to be decreased, and that is only possible if you revise over and over, to that extent that in your last revision that the whole book can be covered maybe in 5-6 hours. So that kind of perseverance is needed. So that means the books need to be revised over and over, maybe 10 times every chapter. And then it will be like all the things, it will be pasted on your mind like some images. So no problem in there. And when you are studying, you should not just mug up the lines, means whatever is written in there. Try to understand, try to read between the lines, right? And now this is about preparation. And another important thing here is the mock test. See, maximum amount of this maximum exams, these are basically MCQ based exams. So there we need to not only MCQ based exams, maybe for ARS mains, which is a written kind of exam. So mocks are very important, because whenever we go for some competitive exam, there will be certain kind of mistake that we are prone to do, some silly mistakes maybe. So those will cleared up, those will rectify itself if you take your mock exam. And maybe in the last months of preparation, in the later period of your preparation, take as much as mock exam you can. Say you can have your question paper from your faculty maybe, or maybe make a group with your friend. So take your friend to make a question paper for you, you make the question paper for your friend, that will also work. So just do some mock exams. And it is not for the what type of questions generally this ARS exam is having, no, that is obviously there, but it is just to sharpen your instincts, your preparation, means you will be exam ready. And then as I also said that do not mug up, but there always there is a need of mugging up some list, some data, some maybe some production data, some yield data, something. So try to form some small easy lines, maybe in your colloquial language, in your native language. Means whenever I study those, means say for Hofmeister series, so that kind of series, I formed something in Bengali, since I'm a Bengali. So maybe you can also form some easy lines from your native language that actually relate with that list, that data. So you can actually remember them very easily, right? So that you need to prepare. Then some data, some chart you can actually paste on your hostel wall, say the MSP of different different crops, different food grains. You can paste it over your wall. Maybe the yields of food grain production, maybe. So you can paste it on your wall. So in that way, you will not have to see, you will not have to read those data or charts on your own. Means very means you do not have to make an extra effort for reading that. Means whenever you are going to sleep, you will that data you will see, and that's it. That is what is needed for clearing this exam. So do this. Then discuss among yourselves, make a study group, ask questions to your friends. So that is very important here, because that shows how much you remember, what is your recapitulation capacity. So that increases. Means actually we have that saying, na, practice means makes a man perfect. So that is that only. And the last month of this exam preparation, be it JRF, SRF, or anything, is very crucial. So you need to be in that zone. Means you need to discuss the subjects uh, very often. Always think of that. See, your mind is only means it has a limitation. See, means storage capacity is very high, but the RAM actually it's limited. So whatever you are thinking, that will be present in your mind. So you be in that zone. Means if you are preparing for agronomy JRF, so study those, discuss those, some list, some data. Forget some other things for some time at least at the that very last month of that preparation. You can organize your notes, materials around some keywords, concepts, and develop a summary sheets actually for course materials. And as I said, the last month is very important. So no new reading material in that last 2 months, right? Whatever the basic books, the advanced books, everything in your syllabus you need to read, reread, revise in that before that thing month, that 2 month. In the last leg of this preparation, only what you can do, only you can revise. No new material you should read in that part. And another thing that I practically follow, and I will also suggest you to follow. See, it is all about now this MCQ type of exam. It is all about the residual knowledge. Means whatever you have read, then you revise. You forget obviously, but you may be forgetting that in your sense, but that is present in your subconscious somewhere. So that information stored in your subconscious mind, maybe that is important for your clearing this exam, right? So do not panic if you're forgetting everything. Means this feeling is very common. I am forgetting this thing, I am forgetting that thing, even I am forgetting the essential plant nutrition, that rule, that law. So that is not it. It is there in your present in your subconscious. So that in the last month, maybe you try to recollect the things, maybe sleep, maybe meditate, maybe. And also just prior to the day of exam, please do not study, because studying in that 1 day 2 day prior to exam, it will lead you nowhere. Whatever information you are gathering from in that period, that will basically overlap with those subconscious things that you have already having there. You need to be calm in those prior to your exam date. You sleep well, at least try to sleep well. Generally it becomes very tense, we do not tend to sleep, but try to sleep well, be calm, but obviously do not go on a trip or party or something. Just calm, be in the zone, be exam ready. That what we call it as a exam ready. So try to do that. So then you will be exam ready. Your subconscious will be very sharp, and you can recollect your these things. So now you have done your preparation. You have given enough of the mock test. No more error you will be doing in that exam hall. So now you are in the exam hall. So what you should do there? Now for MCQ exams particularly, first thing, you should try from the question number 1. You start answering the questions from question number 1. First, means I am giving you a template for answering, approaching each question. First, read the question or statement very carefully. Whatever is written in that question. See, it can be written as rice is a food grain crop. Okay. Or it may be written rice is not a food grain crop. So these are basically two different opposite statements. If we hurriedly go, you can miss that note, and your obviously it will be answered wrong. So first you read that question or statement very carefully, and when you are reading that, you do not see the answer choices, right? You should not see the answer choices, because whenever you are seeing that answer choice, your thinking gets biased. So you did not need that. You should not do that. You read the question statement very carefully. For 1 second or a fraction of second, you try to think about that answer in your head only. So what could be the possible answer? And see, all these things now, all this studying you have been doing, so all this these are present as images in your brain cells somewhere. So some it will lead you to some image. Whatever you are studying, it will lead you to some of that page or some of that list somewhere. It will lead you to that. Then you see the answer choices, and then you will see very confidently you can say that answer number 1 is the right answer. So mark that one. Okay, but after that thinking of that answer of the question in your head, after that you see the answer and read all the answer choices carefully and eliminate the incorrect answer and mark the answer correct. And if you know more than one answer is correct, then consider all of the above. And if the all of the above isn't a choice or isn't the correct choice, then select the best answer, obviously. Now, this is the template for approaching each of the question individually, starting from 1, 2, 3, 4 to the last question. And be calm. You have to give the best shot at solving the problems. And now this template you should follow for every question. And here, if your mind says that this is the correct answer, you tick that one. And this is what we tell as a sure shot ones. So you answered it very carefully. Means answered it very carefully and it is right. So these are sure shot ones. Now forget about those. Do not look back to those questions again. It will only lead you. Means do not, after answering once, do not rethink. Okay, it will only lead you to wrong answer. So do not do that. Whatever comes to your mind first, be sure that is the right answer, and your subconscious, your mind is much smarter than you. You just note that. Then maybe there will be then some difficult questions which maybe after this template also you will be confused between 2 options, maybe 3 options, so that you can't decide in that very instant. So there what you need to do, you mark those difficult ones, but in that first go, do not spend excess amount of time on one question. If it's a difficult one, every question is containing same mark, right? So do not spend time, mark the difficult one and go to the question, next question. Now whatever time is allotted, maybe in the first 60% of that time you should complete that first attempt of all the question paper following the same template. First read the question, do not see the answer, think about the answer, then see the your answer options and mark the one. Following the template for each one, you go to the last question, means up to the last questions, and see sometimes means this is actually happened with me only. Exam, GRF exam, the first 10 questions were from plant science. I am from physical science, so first questions were of general agriculture type and very hard ones. So first questions I don't know, second question I don't know, third question I am confused. But do not lose heart. Just mark them as difficult ones, move to the next one, move to the next one. And actually after 10 questions, all questions are from my field only, but do not lose your nerve. Leave the 10 questions for maybe later part, second attempt. So do not waste time on hard questions in the first attempt. Do not overthink. Then after that first attempt is done, then go to the second attempt. And also, this is my personal choice. Solve the mathematical problems, means keep those one also apart for second, because for some mathematical problems, some calculation if it's involved, then it will require some more time. So keep those one for the second attempt. And then second attempt also you can do, think for a more amount of time and answer some questions. You can, what you can do, you can do the elimination tactics here. So say if you know some say for means if you know some agricultural problem, some agricultural policy implementation, it is around means the question is coming, this agricultural policy is implemented by Government of India in this year. 4 options are there, but you do not know, but do not remember actually. But you know this particular policy is implemented maybe maybe when there is India on the World Cup. So that is around 2011, that we all know. And there is a answer which is having an option of 2012. So you mark that one. So that is called elimination technique, right? So that way you can also eliminate the incorrect answer and mark the right ones, even if you do not know the right answer there. So do that. And maybe also doing this all kind of stuff, that second attempt, some question will also be left. So mark them for the third attempt. That third attempt, I am talking about maybe 10-15 minutes in your total examination time in the last leg of that exam. So there it is time to take some calculated guess. Now, it is not wrong to have a calculated guess. Take it from me, some might say that only you should answer the sure shot ones. No, if you study hard, your calculated guess means, in Bengali there is a word called tukka. That means this calculated guess only. And it actually says MCQ exam में pass होने के लिए तो कोई भी हो जाता है. नहीं. It actually shows the amount of preparation that particular student actually had done. So his calculated guess will be very pin to the point, actually very pinpoint guess you can make. And there all that residual knowledge present in your subconscious mind actually come into play. And whatever you have read in those 1 year, whatever you maybe read in your UG courses, so everything you try to remember, make some calculated guess, means anything, your mind is very smart. It will give you some hints. Follow that hint and mark the answer. Remember, in any case, nowadays, in our time, it was 0.25 for wrong answer and 1 for right answer. Nowadays, I think it's 0.33 for 1 wrong answer. So see, after your sure shot ones are done, those maybe 85 answers, those 85 marks you are having. Then you take 4 chances, right? 4 chances may, if 1 is right and like 3 is wrong, then you are no gain, no loss, means no problem. But even if your 1 calculated guess extra is right, then you are having like 1 marks. That is good, na? So do not be afraid to take those calculated guess, but do those only in the third attempt, not before that. Okay, and try not to take breaks in that exam period because time is precious, and do not lose your focus. And this is for MCQ. If we talk about the descriptive questions, there for preparation actually, there is also mock is very important. Formulate probable questions during your study period, means maybe collect old question papers, take help from your faculty, from your seniors who are already been in that system, who have cleared the exam. So request them to make some question paper for you, and solve as many mock test papers as you can. And then actually this question making is not very difficult. Means whenever you are studying some particular chapter, you take a white sheet and write what can be the probable question that can come from that. Now that is now this is not for making out like some very ideal answer. No, some notes. No, nothing like that. You just formulate question. That's it. And maybe after maybe once a month you can just hurriedly select some question papers, or maybe tell anyone else of your friends, just hand over that paper. There let's say 50 questions are written from one chapter. You just ask him, just select number. So he select some random numbers. He selected like 5, 6, 10, 12, like that. So that is your paper for that day. And divide some times and allot times according to marks. See, you know your exam schedule, and you know how much time is allotted, the total time. So say if 12 questions are there and 1 hour is there, so that means 5 minutes a question. So in that time, means in that question paper, the time is very important. It's just like gold there in question. exam hall. So here also, like the MCQs, do not skip a question. First question, second question, third question, fourth question, like that you just go on. And here that difficult question marking, do not do that. Per question you have 5 minutes. Write the answer for 5 minutes, move to the next question. Do not stop, do not overthink. Whatever comes to your mind, write that and move on. See, if you are having like 100 means like 100 questions there, so each of the question is having like 5 marks. So for assuring 5 marks in one question, you should not lose like 2-3 marks or more than means 5-10 marks for some other easy questions which actually you know the answer to. So approach each question individually, allot times also for individual question, 5 minutes per question. Whatever you can recollect, you can write, but within that 5 minutes. 5 minutes over, move to the question, next question. Whatever done is done. And so that means you need to pack up a lot of information in that 5 minutes only. And in the ARS mails, what they are doing these days, they are basically giving you a very long paper, means both pages you can write actually. So do not try to fill up those pages. No, try to write as much as you can by that 5 minutes, right? So you need to pack a lot of information in that 5 minutes. So try to answer in diagrams, figures, like the important words, the keywords that you should write. Say it is asking about you some nutrient cycle, maybe potassium cycle. So you can write 1 paragraph, 2 paragraph of potassium cycle, but it will give you 5 marks only, not more than that. But instead of, if you can just make a diagram, means this pool, that pool, that pool, plant uptake, fertilizer input, then to organic matter, whatever, that for making the diagram you will need much less time. Just clearly label that diagram. It will also fetch you 5 marks, because the person who will be evaluating this that answers, he sees the diagram, he knows that this student actually know all this stuff, so give him 5 marks. So try to answer in diagrams and figures. Now do not there is no means do not do this, means you are also making a diagram, figure, and also writing that about that diagram. Do not do that. That diagram, clearly labeled diagram or figure, is good enough as your whole paragraph. And no marks for beautification, means 1 pencil for graph, 1 blue pen, 1 green pen, 1 black pen. No, it's just a waste of your time. Nobody needs that beautification. What you need is the correct answer and clear answer with a good handwriting, at least that is understandable. That is enough. So do not waste time on that, right? And again, do not think and then write. Try to think and write, means it will save you time. Now this is actually what takes practice, means thinking, recollecting, and writing. So this is not a writing speed. Means हम बोलते हैं ना कि this guy has a very very good writing speed. So it is not a writing speed. It is basically how fast you can recollect from the memory what you have read and put it in effective words in your answer sheet. So that takes lots and lots of practice, and mock tests are the key there. Mock test, how the many mock tests you will give, you will be more what I can say, more you will be an artist in that field actually. So then again, when you are preparing for a descriptive exam, do not try to read the same topic from different books. One standard book is enough. See, for as I made mention actually, all this thing you are studying, it will store as a memory, and human memory stores information in the form of image. Say I am studying nitrogen today. I am studying nitrogen from the Indian Society of Soil Science book and also from the DKS book. So then whenever the nitrogen comes to my mind will be having 2 pages. images of 2 pages, one from this book, one from that book. So then it is taking is overlapping images, and I may get confused, and the recollection of that memory, that particular information, will be somewhat hazy, somewhat not very effective. But if I have read that thing only from one book, so whenever I see the word nitrogen, say this fundamentals of soil science, that page comes to my mind, and I can write it, because that image will be very clear, right? One image is always very clear than overlap of 2 images, that we all know. So that is why when you are preparing for a descriptive exam, maybe for mains of any this ARS kind of thing, so do not try to read too many books. Try only the standard books. One topic from one standard book is enough. So that is how you approach this thing. And again, what I already told you, do not overthink and do not take 1 second extra for a question. Perfection in one difficult question may cost you the next easy question. So that do not do. And do not leave any question unanswered. Anything is better than 0 marks. We all know that, right? So whatever comes into your mind, you just write. And if you have prepared well, you are studying agriculture for 4 years, then again 2 years, again 3 years. So you know it is not a rocket science. You know everything about the agriculture. So you can write something, and that is better than nothing. And completion in time and quality of information is important, and not the volume of words. So do not try to write meaningless lengthy paragraphs. Be responsive, active, and alert in those exam hours. Adjust your daily routine according to at least 1 month prior to exam. Now this is very important. What happens with us students, actually, some of us have very peculiar reading schedules, actually, studying schedules. At least for me, I used to study in the late night hours, means like 12 p.m., 6 a.m. I am studying, and in the morning 10 a.m. I am sleeping. But that routine actually needs to be shifted around your exam this time. So means try to be very alert, very responsive in that particular time period of the day, at least for 1 month prior to your exam. So then you will be your best in that exam hall, and you can actually give better answers. And again, no last minute studying, and try to sleep well in those in that time, and it improves your thinking capability also. And if you have selected for this thing, then you need to prepare for interview. And for interview, there is some tips, actually I try to share with you, that for interview, whoever people are interviewing you, they are very intelligent. They are in this field only for 40-50 years. So do not try to fool them. And in that interview panel, nobody is trying to judge your knowledge, because that knowledge has been judged in your prelims, in your mains exam. So in that panel, he is basically studying, if we means if we select this particular candidate, whether he will be justifying with the salary, he will be given, whether he will be truthful and ethical. So that things he will be judging there. So, and obviously you are not supposed to know everything, and your knowledge cannot be judged in 10 minutes. So it is basically, it will judge your smartness. It means, say some problem is being thrown at you, and you should have that problem solving attitude. Means there is two kind of attitude. One problem is thrown at you, and you tell, sir, mujhse na ho payega, I can't do that, I have this problem, I have that problem. No, that is not it. In Indian situation, I throw you a problem, you have to tell me some solution, at least try to solve that. So that is what we call smartness. So that attitude is generally judged in that interview panels. And obviously be polite, but not be nervous, and be confident, but not overconfident. That means, can you solve that problem? Yes, sir, this problem is very solvable, I can solve it. No, not like that. It is a problem. Try to means see that seriousness of that problem. Try to assess that problem very critically. Then pass your judgment, sir. I can do that. Maybe this can be done, but after thinking. So that kind of stuff. And in many interview, they actually ask about, please tell me about something yourself, maybe a CV you can write. For, in fact, for ARS, they generally ask for one CV. So every word of that CV should be thoroughly thought upon, because if I am going for an interview, the interviewer do not know me. He will know me by that CV only. So whatever I write in that CV, that is the points you will be taking in mind for asking questions. See, say if I have written in my CV that my specialization means I am doing my PhD in carbon dynamics, maybe in conservation agriculture. So that means that word he then read, acha, carbon dynamics. Then he can ask me anything about carbon. Conservation agriculture, anything about conservation agriculture, zero tillage, where it is practiced, how much it is practiced, even how much is the price of your this happy seeder, that he can also ask. So all that things, whatever you are writing in your CV, that is very important. Or maybe you are making a PPT there. So those things that you are actually giving points to the interviewer for asking you questions. So always try to bring your interviewer in your comfort zone, in your playing field. Then only you can be answering all those questions. Do not even go to their playing field, because he may be means a specialist maybe in clay mineralogy. You do not know much about clay mineralogy. So if he asks about clay mineralogy, you will not be able to answer many questions. So try to bring him to your field. And even there also, even hobbies do matter, because I have some instances, not happened with me, but with some of my colleagues there, some of my friends. So if someone tells na okay, my hobby is like reading books, so he may ask what is the last novel you have read, and please try to tell the story in 2 lines. So he can ask. So do not lie there. So that is very useful there. Do not lie there. Do not write anything there. Say one I've written playing cricket is my hobby. So the question comes, what is a Chinaman bowler? So that kind of things also happen. So try to read those and be confident. So that is all about this. And be presentable. Do not excessive makeup or wear formals. And also have a clear idea about the organization you are applying for and your role in it. Have an overall idea about the subject. And the interviewer actually will ask questions according to your specialization and the job you are applying for. So keep that thing in mind. So that is all about this thing. And thank you for inviting me. I think this will be helpful. And you may contact me in my mail ID, my mobile number, and you can follow this. Means there are several YouTube channels also. One is by Dr. Vipin Kumar, my colleague only. So you can see there are some videos, the strategies of different people who are actually successful, who have gone through this process and be successful. So try to see from their experience. So best of luck and thank you. Thank you, sir.
Thank you, sir. Thank you very much. If among the participants having any queries, you can ask.
Hello.
Birla ji.
Yes.
Sir, what is the role of STO?
STO.
What is the role of?
STO, sir, ARS STO का तो इस बार
STO means उसका क्या full form क्या है?
नहीं सर, उसका वर्क क्या है? जैसे आपने बोला कि मतलब इंटरव्यू में पूछ सकते हैं कि
Exactly. तो टेक्निकल ऑफिसर है ना STO.
यस सर.
Yeah, so टेक्निकल ऑफिसर इसका वर्क क्या होता है? Generally जैसे I am a scientist, so I am having some projects here according to our institute mandate. So टेक्निकल ऑफिसर का होता है कि उसी प्रोजेक्ट में if I instruct him to do something, maybe I will instruct him to जाओ भाई आज हम ना ये एक हम ट्रायल प्लान करेंगे. So my responsibility is planning of that experiment, to oversee that whether this is happening or not. But that technical he needs to see whether that 50 kg nitrogen has been given at that very particular date. So this is for a maybe a technical in agronomy. But if that STO is in soil science, then उसको ये पता होना चाहिए मतलब डेली वर्क कैसे हमें करना है, मीन्स some of incubation study, maybe procurement of the chemicals. So everything comes in that STO, and more. This is the post is of technical officer, so he should have the technical knowledge of that subject, right? And syllabus is same as ARS, so more of that. So that is what I can say. And this actually varies for means what subject you are asking from, agronomy.
एग्रोनॉमी अभी इंटरव्यू होने वाला
हाँ,
है ना,
तो
तो इसलिए होगा।
हाँ, तो एग्रोनॉमी में क्या होगा ना, एग्रोनॉमी में मतलब फील्ड कैसे प्रिपेयर किया जाता है, कौन सा आप जहां के लिए अप्लाई कर रहे हो वहां पे कौन सा पर्टिकुलर क्रॉप इंपॉर्टेंट है, उसके डोज क्या है, फिर फील्ड जब जब हम कोई ट्रायल लगाते हैं तो ट्रायल में इवन फील्ड लेवलिंग कैसे करते हैं, उसमें से फॉर सी अगर हम कर रहे हैं कॉन्वर्सेशन एग्रीकल्चर, तो उसके लिए पर्टिकुलर अमाउंट ऑफ पर्टिकुलर टाइप ऑफ ट्रैक्टर मतलब वो लगते हैं, हमारे हैप्पी सीडर लगते हैं, टर्बो सीडर लगते हैं, तो उसका प्रिंसिपल क्या है। फिर ऐसे छोटे मोटे चीज मीन्स यू नीड टू सी, मीन्स इफ यू आर अ स्टूडेंट ऑफ एग्रोनॉमी इन योर यूनिवर्सिटी, यू जस्ट गो टू दोस फील्ड्स और वहां पे क्या काम हो रहा है, सोइंग कैसे होता है, हार्वेस्टिंग कैसे होता है, कैसे इवन आई हैव सीन स्टूडेंट्स who worked means who have completed their thesis but they do not know how to take grain yield weight. ऐसे नहीं होना चाहिए, कितना मॉइस्चर परसेंटेज होना चाहिए जब आप यील्ड का डेटा लेंगे, ठीक है, कैसे लिया जाता है, ग्रीन सीकर का वैल्यू आप कैसे लेंगे, ठीक है, प्लांट हाइट कब मेजर करना होता है, कैसे प्लांट का गर्थ मेजर करेंगे, ये सब चीजें, तो ये सब करना पड़ेगा, तो ये चीज जानना भी पड़ेगा फिर, ठीक है। फिर इसमें अगर आप इंटरव्यू देने जा रहे हैं तो आई डोंट नो व्हाट इज योर मीन स्पेशलाइजेशन, मतलब आपका क्या स्पेशलाइजेशन है, आप एमएससी में हैं?
यस सर, पीएचडी में,
पीएचडी में, तो आपका आपका एमएससी हो गया होगा, तो एमएससी का टॉपिक क्या था, रिसर्च टॉपिक?
सर इफेक्ट ऑफ सिलेज एंड वीड मैनेजमेंट प्रैक्टिसेस ऑन ग्रोथ एंड यील्ड ऑफ लेंटिल।
ओके ग्रेट, तो आपने तो टिलेज पे आ गया। सो आपको पता है कि स्ट्रॉ मैनेजमेंट सिस्टम क्या होता है?
स्ट्रॉ मैनेजमेंट सिस्टम
क्या होता है
सर? स्ट्रॉ के मैनेजमेंट के लिए उसको मल्चिंग में यूज कर सकते हैं।
इट्स अ इंस्ट्रूमेंट पार्ट, यू डोंट नो अबाउट इट, राइट?
नो सर।
तो यू नीड टू नो, इफ यू डोंट नो यू नीड टू नो। जैसे आपने जीरो टिलेज पे किया, तो जीरो टिलेज पे कौन सा सोइंग इंप्लीमेंट है, हैप्पी सीडर है, टर्बो सीडर है, पहले जीरो टिल सीड ड्रिल भी था, तो वो कैसे डिफरेंट है नॉर्मल टिलेज से, ठीक है, वो इंस्ट्रूमेंट के बारे में जानना होगा। फिर कॉम्बाइन हार्वेस्ट, एक्चुअली ये सब आता है जब हम कॉम्बाइन हार्वेस्टर से हार्वेस्ट करते हैं, उसके पीछे एक कटर टाइप का सिस्टम लगता है, उसको हम एसएमएस या स्ट्रॉ मैनेजमेंट सिस्टम बोलते हैं।
ओके सर।
तो उसके बारे में आपको जानना पड़ेगा वो कैसे वर्क करता है, और इवन इफ ये जो इम्प्लीमेंट है ना, ये कितने का आता है, और इसको अगर कस्टम हायरिंग करे तो हाउ मच कॉस्ट एक का पर डे पर एकड़ हिसाब से आता है, तो दैट थिंग आल्सो यू नीड टू नो। एंड आप जब सोइंग करेंगे, आप जैसे नो टिलेज पे काम किया आपने, तो मैं अगर आपको बोलूं कि नो टिलेज पे सोइंग किया और टिलेज करके सोइंग किया, तो उसमें टिलेज ऑपरेशंस में कौन सा डिफरेंस है, मतलब एक में रोटावेटर लगेगा एक में नहीं लगेगा, तो कितने बार रोटावेटर चलाना है, किस में नहीं चलाना है, कितने में, अब इसमें जैसे आप फर्टिलाइजर अप्लाई करेंगे वो भी कब करना है, कैसे करना है, साथ में करना है सीड के या फिर पहले करना है, अब जो सीडिंग डेप्थ है वो भी कितना होना चाहिए, ये भी पता होना चाहिए। तो ऑल दिस थिंग्स यू शुड नो। आई थिंक यू हैव अ, मतलब मैंने आपको शायद कुछ तो आइडिया दिया होगा आपको कैसे अप्रोच करना चाहिए।
यस सर।
ठीक है, मतलब नॉट वो ओनली नहीं जो आपके थीसिस में लिखा है वो तो पता है सबको, आपने किया है बहुत बढ़िया, बट एक्चुअल में फील्ड में क्या है वो चीज आपको करना है,
प्रैक्टिकल नॉलेज,
आगे जाके आपको, हाँ प्रैक्टिकल नॉलेज, आगे जाके अगर आपको एक एक्सपेरिमेंट लगाना होगा तो ये सब चीज आपको ही फाइनल करना होगा और आपको जाके ओवरसी भी करना होगा, तो यू नीड टू नो।
ओके सर, थैंक यू सर।
या थैंक यू।
Anybody?
और कोई anything else?
I think nobody is there. Mr. Gule, please conclude the session.
Yes, sir. We are very much thankful to you, sir, on behalf of Agriculture Extension and Communication Department, PGI MPKV Rahuri, you have really given a memorable lecture in today's session. In that you have very nicely and deeply explained the site-specific nutrient management concept, fertilizer recommendation based on different concepts and approaches. Then methodology for developing basic data on needed parameters for targeted yield. Then formulation of adjustment equation for without and with FIM. Then you have also explained the nutrient expert decision support tools for precision nutrient prescription, and that normalize the difference vegetation index with green seeker handle crop sensor, then leaf color chart, Android based mobile application for real time nitrogen management for wheat and rice. And finally, tips for preparation of different exams. Sir, you told very admirable tips for preparing short answers and brief answers, and how to read the books and how to revise the books. Actually, I very appreciate to you, sir, because you have given very nice tricks from very small things, which are which students should be always remember that for obtaining success in exams. And finally, given very nice tips for the facing interviews if a student selected. So thank you once again, sir, for giving your valuable time and guidance and connected with us. Thank you so much, sir.
Thank you, Abir sir.
Thank you,
Very
sir,
nice
for
session is
inviting
there.
me. Thank you.
We please, sir. Okay, okay. Thank you.
Shall we conclude session, sir?
Yes, 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 yes.